Would you start?
Yes, hi there. Yes, so it will say Jonathan Statham in your programmes, but unfortunately he couldn't make it, so I'm standing in for him. I wasn't actually involved in this project, so if any of you do have further questions about the project, I can give you his contact details for any further questions. So I am a farm animal vet in practice in the UK, working for Bishopton Veterinary Group in North Yorkshire. But we do have our sister company, which is very much involved in veterinary-led industry work. There are 4 divisions to the company. So RAF stands for Research, Advanced Breeding, Food Futures Consultancy, which is where this project lies, and Training. So what is this project? So we work together with our partners, including FERA, to develop a loop-mediated isothermal amplification assay for some of the major meat-borne pathogens, such as Salmonella, Campylobacter, and E. coli 0157. We wanted to develop a simple and rapid testing procedure which was amenable to automation, which required few steps that could be carried out by lay staff in the abattoir with minimal training. We also wanted to create a system which meant that multiple tests could be performed on one single sample. So this is important because currently in the UK, when we're testing for various pathogens, results can be retrospective of when meat has actually left the food plant. So for example, with Salmonella testing currently in the UK, samples may be taken 2 weeks prior to slaughter. So this doesn't account for any post-sample contamination. And further to that, samples are then repeated in the abattoir, but results take a couple of weeks. And by this point, food could already be on the plates of humans, which obviously can result in very expensive product recalls and disease. So loop-mediated isothermal amplification of DNA. So it's isothermal, which means it's cheap to run. It's also very rapid at testing, and results are within 1 hour. It's robust. So this procedure is less sensitive to the inhibitory substances that PCR might be susceptible to, and there's also no need for DNA purification or enrichment. It is highly specific, and it avoids interference by non-target DNA that might be present in the sample. So what did we do? So in poultry abattoirs, pig abattoirs and cattle abattoirs, surface swabbing was performed on carcasses as per the United Kingdom Food Standards Agency standard procedure. So this involved a sterile saline soaked swab, which was rubbed down the side of the carcass. 2 swabs were taken at the same time. The carcass selection was completely random, and also the side of the carcass that was swabbed was completely random. The carcasses were swabbed after they had passed meat inspection, but before chilling. So after the swabs had been taken, the swab bag actually contained 10 mils of 0.9% sodium chloride solution, and the swabs, after transporting to the laboratory, were squeezed repeatedly for 2 minutes in the bag before undergoing cell lysis for 5 minutes at 85 degrees Celsius. They were then transferred to the LAMP instrument for further testing. In order to validate the test, in conjunction with the samples going off for LAMP analysis at the FERA laboratory, they also went to the FSA laboratory for culturing, so that we could compare the results. So I'll talk a little bit about the Salmonella results that we got. So when generic Salmonella was tested for, all Salmonellas were detected. However, with the test actually currently only detects typhimurine and enteritidis in terms of individual serotypes at the moment. So these are some of the results that were found, and as you can see, the results were all within 1 hour. That also means that if a sample is taken, if the sample is positive, at that point it can actually be held at the abattoir for further testing if necessary. Some further results there. So in terms of sensitivity, we found that for cattle swabs, the lamp assay is able to detect Salmonella when the swabs have been spiked with 10,000 CFU of Salmonella for both generic Salmonella and Typhimurium and Enteritidis. However, with turkeys and chicken swabs, we found that it was able to detect a level of 1,000 CFU per swab spiking. So what could be the future benefits of this test? So rapid results within 1 hour, it goes without saying. Also simultaneous detection of species through one sample collection. Also any sample type can be used, so whether you're swabbing carcasses, boots, meat samples or the environment. No DNA isolation is required, so this means that anybody who has minimal training is able to perform the test, which means that fewer staff will be needed. So potential uses, it can be used in factories for monitoring, during meat inspection and also enforcement activities. There is also the possibility of border import controls and environmental health. Thank you for listening. As I say, I can attempt to answer questions, but as I wasn't involved in the study, I may need to forward you on to Jonathan.
Okay, thank you. So.